Hello and welcome back to G Fun Facts Online. Today we are diving deep into a topic that sounds like it was ripped straight from the pages of a science fiction novel, but it is actually happening right now in fields and beehives across the world. We are talking about the end of chemical warfare in agriculture and the rise of biological precision. Imagine a sniper so precise it can find a single enemy soldier in a crowded stadium and neutralize them without disturbing a single spectator. Now imagine a pesticide that works the same way. It can drift over a field of blooming flowers, coating the petals and leaves, and yet, when a honeybee lands to sip nectar, it is as harmless as morning dew. But when a destructive beetle takes a bite, it triggers a genetic kill switch that stops its heart. This is the reality of RNA interference, or RNAi. It is a technology that is poised to replace the century-long era of toxic sprays with an age of genetic intelligence. To understand why this is such a monumental shift, we first need to look at the crisis we are currently facing. Modern agriculture is fighting a losing war. For decades, we have relied on neurotoxins and broad-spectrum poisons. These are the sledgehammers of the farming world. They kill the pests, sure, but they also decimate the beneficial insects that keep our world alive. We are seeing a phenomenon known as the resistance nightmare. Pests are winning. Take the Colorado potato beetle, for instance. It is a notorious destroyer of crops. This beetle has developed resistance to over 50 different classes of chemical insecticides. Farmers are forced to spray more often and with more toxic cocktails, yet the beetles keep eating. Then, there is the pollinator collapse. We are losing our bees. A toxic soup of stressors is driving colony collapse disorder, and chemical pesticides are a primary culprit. Neonicotinoids, which were once hailed as safe, are now known to disorient bees, making them unable to find their way home. And finally, there is the environmental toll. Traditional chemicals persist. They leach into groundwater, linger in soil for years, and bioaccumulate in the food chain. The world does not need stronger poisons. It needs smarter ones. This is where RNA interference comes in. Now you might be thinking this is some scary synthetic chemical invention, but it is actually a natural biological process that already exists inside your cells, in plants, and in insects. It was discovered in 1998 by Andrew Fire and Craig Mello, a discovery so profound it earned them the Nobel Prize in Physiology or Medicine just 8 years later. So how does it work? Let's break it down. DNA is the blueprint of life, but messenger RNA, or mRNA, is the contractor that reads the blueprint and tells the cell what proteins to build. If DNA is the cookbook, mRNA is the handwritten note telling the chef to bake a cake. RNAi pesticides work by introducing double-stranded RNA that matches a specific sequence of the pest's mRNA. When the pest eats the crop coated with this specialized RNA, its cells recognize the double-stranded structure as a viral invader. The cell's defense system, utilizing an enzyme called dicer, chops this RNA into small pieces. These pieces then guide a complex called RISC to hunt down and destroy any mRNA that looks exactly like them. In simple terms, the pesticide intercepts the note to the chef and shreds it. The result is that the pest's cells stop producing a vital protein, perhaps one needed to digest food or maintain cell structure. The pest stops eating and dies, often within days. The true magic of this technology lies in its specificity, something we call the genetic barcode. Every species has a unique genetic code. The sequence of mRNA required to build a stomach enzyme in a Colorado potato beetle is different from the sequence in a honeybee, a ladybug, or a human. Scientists designed the RNAi spray to match only the pest's unique sequence. For the pest, it is a lethal instruction to shut down life support. For the bee, it is just a string of nucleic acids, essentially nutritional noise, that is digested like food. It has no effect because the password does not match the bee's genetic system. Perhaps the most emotional and critical application of this technology is in the fight to save the honeybee itself. For years, beekeepers have been battling the Varroa destructor mite. This is a parasitic arachnid that attaches to bees and sucks their fat bodies, weakening them and spreading deadly viruses. To fight them, beekeepers currently have to put miticides directly into the hive. These are harsh chemicals. They can harm the bees, contaminate the honey, and eventually, the mites develop resistance to them. But a company called Greenlight Biosciences has developed a game changer. They created an RNAi product with an active ingredient called Vadiscana, specifically to target Varroa mites. It is a syrup placed in the hive. The bees eat the syrup and feed it to their brood and the mites. Here is the brilliant part. The bees possess enzymes that break down the RNA quickly, so it does not hurt them. However, the mites absorb the double-stranded RNA, which targets a gene essential for their reproduction and survival. The mites die or become sterile, while the bees remain completely unharmed. Field trials have shown that this treatment can reduce mite populations significantly without the toxicity associated with chemical strips. It breaks the cycle of the vampire without poisoning the host. This represents the first time we have used a pesticide inside a beehive that is biologically incapable of harming the bee. But let's move from the hive to the field. While saving bees is noble, the agricultural industry runs on crop yield. The proof of concept for RNAi as a commercially viable tool came with a product called Calantha, the first foliar RNAi bioinsecticide approved by the US EPA in January 2024. Its target is the Colorado potato beetle, that pest we mentioned earlier that strips fields to bare stems. Calantha is sprayed just like a normal pesticide, but its performance is revolutionary. In field trials, it performed as well as leading chemical insecticides. However, unlike chemicals that might persist for months, Calantha degrades in the soil within 3 days. If a bird eats a beetle killed by Calantha, or if a farmer breathes in the spray, the RNA degrades harmlessly. It targets a gene sequence only found in the beetle family. This product proved that RNAi could be affordable and effective at an industrial scale. While sprays are one method, another approach is engineering the plant to produce the RNAi itself. Bayer's SmartStax PRO corn is a prime example. This genetically modified corn does not just produce the traditional Bt toxin, which is a bacterial protein toxic to pests. It also produces double-stranded RNA targeting the corn rootworm. When the rootworm larva bites the corn root, it ingests the RNA produced by the plant. This turns off a gene essential for the worm's life. This matters because the corn rootworm is known as the billion-dollar bug, costing farmers immense sums annually. It had begun to develop resistance to traditional traits, but the addition of RNAi provides a completely new mode of action, effectively breaking the resistance cycle. The environmental benefits of this shift are difficult to overstate. First, there is biodegradability. Chemical pesticides often contain forever chemicals or heavy metals. RNA is a biological molecule. In the environment, whether in soil or water, bacteria and UV light break it down into simple sugars and phosphates within days. It leaves no toxic residue. Second is soil health. Earthworms, nematodes, and soil bacteria are vital for healthy crops. Broad-spectrum fumigants kill them all. RNAi targets only the pest, leaving the soil microbiome intact. And third is worker safety. For farm workers, the risk of acute poisoning from handling these products is virtually non-existent compared to organophosphates. Historically, the barrier to this technology was cost. Making RNA used to be incredibly expensive, costing thousands of dollars per gram in the lab. This made it impossible to spray on a field. But companies like Greenlight Biosciences solved this by developing cell-free manufacturing processes. They essentially created large fermentation vats, very similar to brewing beer, where yeast or bacteria churn out massive quantities of double-stranded RNA at a fraction of the cost. Production costs have dropped to under $1 per gram, making it competitive with premium chemical pesticides. The market for RNAi pesticides is projected to grow from a niche sector to a multi-billion-dollar industry by 2030, driven by mandates like the European Green Deal to reduce chemical pesticide use. The revolution won't stop at insects. Researchers are looking at weed control next. Weeds are harder to target because their cells are protected by thick, waxy coatings that are hard for RNA to penetrate. However, companies are developing surfactants and nanocarriers to slip the RNA inside weed leaves. If successful, we could have a spray that kills Palmer amaranth, a superweed resistant to Roundup, without harming the soybeans growing right next to it. Fungicides are also on the horizon. RNAi sprays are being developed to target gray mold on strawberries and grapes, potentially replacing copper and synthetic fungicides to ensure our wine and fruit are free of chemical residues. Of course, no technology is perfect. RNAi faces its own set of hurdles. Public perception is a major one. Because this involves genes, there is a risk of consumer confusion with GMOs. It is vital to communicate that sprayed RNAi does not modify the genome of the plant or the pest. It is a temporary silencing effect. There is also the risk of resistance. Just as pests evolved resistance to chemicals, they can theoretically evolve resistance to RNAi, perhaps by changing their gut chemistry to destroy RNA before it enters their cells. Integrated pest management will still be necessary. And finally, regulation. Regulatory bodies worldwide are still writing the rulebook. The US EPA is leading the way, but strict regulations in places like Europe could slow adoption. In conclusion, we are witnessing the end of the sledgehammer approach to farming and the beginning of the scalpel. RNAi pesticides offer a vision of agriculture where we work with the biological code of nature rather than against it. For the beekeeper, it means hives that thrive without fear of mite infestations. For the farmer, it means crops protected from superpests without the fear of resistance. And for us, the consumers, it means food that is grown with intelligence, precision, and a profound respect for the ecosystem that sustains us. The genetic sprays are here, the pests are silent, and the bees are buzzing louder than ever. Thank you for listening to G Fun Facts Online, and we will catch you in the next episode.